In this video, we will be discussing about the SOS response system. So what do you mean by SOS response system? This SOS response system is activated when a cell requires a repair system. So there are various type of damage which are created in the DNA due to some of the mutations as well as due to some of the UV radiations. So there are various type of repair mechanism, and when this repair mechanism cannot work or it cannot repair the DNA, during this time the SOS response system work for activating some of the genes which are involved in the repair mechanism. So in this SOS response, there is involvement of two genes. One is a positive regulator, another one is a negative regulator. So the REC-A protein which is present, it is a positive regulator for this SOS response, and the LEX-A protein which is present, it is a negative regulator. So this happens in case of a prokaryotic cell, and in the prokaryotic cell, the genes which are the structural genes which are present, they will be synthesized as a mRNA. First, there is a gene which is present in the DNA. From there, during the process of transcription, there will be formation of mRNA, and during the translation, the protein will be formed. So, in case of a prokaryotes, we have a operon system. In the operon system, first the promoter is present, then the operator is present, next comes the structural genes. So, here the structural genes are the repair proteins which will be activated by the SOS response system. What happens in case of a normal shell or during the normal transcription and translation process? Here the RNA polymerase comes and bind, and it crosses the promoter as well as the operator region, and the structural genes are synthesized into the mRNA. Then by the process of translation, a protein is formed. Here in this promoter region, the negative regulator LEX-A is protein. LEX-A protein is present. So what was the work of this LEX-A protein? It was the inhibitor of operon system. As it is the inhibitor of this operon system, it will inhibit the work of REC-A, UMUC as well as UVRA. These were the structural genes present in this operon system, and the LEX-A will be present in the promoter region. This blue one is the promoter region, and here there is a box which is known as SOS box. Upon this SOS box, there is a LEX-A protein which is present, which is inhibiting the operon system. So here when the RNA polymerase will come and bind, it cannot cross this portion. So there will be no synthesis of the structural genes in case of a normal shell. But when the cell or when the DNA is getting damaged, it will activate some of the proteins, that is the REC-A protein. In the normal case, the REC-A protein is in the inactivate form. We can see here that in the normal shell, the REC-A protein is in the inactive form and the LEX-A protein is in the active form. That means it is inhibiting the operon system, because there is no need of SOS repair system, or SOS does not need to activate the repair enzyme or the repair proteins, because the cell is in the DNA is in the normal condition. It is not in the mutated condition. So it does not require a repair mechanism. But what happens when a cell is getting damaged due to some of the UV radiation as well as some of the mutation? There we require some of the genes which are responsible for the repair mechanism. So during this case, the REC-A protein which was inactive in the normal condition, it will be activated. When this REC-A protein is activated, what it does, it breaks the LEX-A. The LEX-A protein is having a autocatalytic property. So this REC-A, what does it induces the LEC-A to activate its autocatalytic property, due to which the LEX-A is degraded into two parts. So now what happens here in this region where the SOS box was present, and it cannot bind in that region because the LEX-A is degraded now. So the RNA polymerase which is present, it can now synthesize all the genes which were responsible for the repair mechanism. That means the operon system is activated now. So what happens in a shell? In the normal condition, the REC-A protein is in the basal level. So the LEX-A protein which is present, it can be degraded by this REC-A protein. After that, the REC-A protein will be activated when the RNA polymerase will synthesized into mRNA as well as protein. So the REC-A protein which is present here, it will be involved in the recombination repair, and this REC-A protein along with degrading the LEX-A protein, it will degrade the UMUD. So this UMUD will be cleaved and it will be forming UMUD dash. Two UMUD dash and UMUC will be responsible for translation synthesis. In this part is known as polymerase 5, which is responsible for the repair mechanism or translation synthesis. We will see what do you mean by a translation synthesis. So now another gene which is present is the UVRA. This UVRA along with the UVRB, it is responsible for the nucleotide excision repair. So now let's talk what about the translation synthesis. What happens in a normal DNA? There will be formation of dimer, that means TT dimer. So in this case, what happens of DNA polymerase which is present, it cannot cross this portion because it is bulky now, it cannot cross easily. So during this time there is a translation synthesis or translation type of repair system is activated. So what it does, it replaces this process and the RNA polymerase will come here and synthesize the genes which are present. It will bypass this portion, and this is done by polymerase 5, which is a mixture of DNA UMUD dash as well as UMUC. Here UMUD dash are into two UMUD dash are present and one UMUC is present over here. Now let's see this diagram. What happens when there is no DNA damage? Suppose there is no DNA damage, during that case the SOS response is repressed. That means in the SOS box which is present in the promoter region, and in it the LEX-A dimer is present. That means it is bounding in the SOS box, and the RNA polymerase will not synthesize these proteins, and these proteins are regulated by a LEX-A protein. So when the DNA is getting damaged, the replication for styling occurs. Here that there will be synthesis of a DNA polymerase, but when there is some of the damage, the DNA polymerase 3 cannot work. So there is a stack over here. When the DNA is replicating or it is transcribing or translation is taking place, there will be a single stranded DNA will be present here. When the single stranded DNA is present, the REC-A which is present in the basal level, it will be activated more. So what it does is the REC-A mediated LEX-A cleavage takes place. Here the dimer which was binding in the SOS box, it will be cleaved into two subunits. Then the SOS box will be activated, or the RNA polymerase now can synthesize the LEX-A regulated gene, and there will be a response mechanism to activate the genes which are required during the DNA repair process. So this is all about today's video. Thank you. If you like this video, please do like, share and subscribe.